It's Dr. Smeets from St. Vincent's Institute in Australia.
Yeah, good morning. Before I start, I would like to thank the organizers for giving me the opportunity to present this new mouse model of MDS. And I've got nothing to disclose. So SRSF2, or serine arginine-rich splicing factor 2, is a component of the spliceosome. It functions in pre-messenger RNA splicing by binding to the exonic splicing enhancer sequences with its RRM domain and to other spliceosome factors with its RS domain. It's important to mention that spliceosome mutations, including in SRSF2, are mutually exclusive and only occur as heterozygous events. They are also associated with distinct disease phenotypes and outcomes and exhibit specific patterns of co-occurring mutations. So SRSF2 mutations. So in the human SRSF2, P95H is a common hotspot for mutation, and with the most common mutation, probably 95 to histidine mutations. And this histidine mutation results in a change in exon inclusion in about 1% of the total splicing events. SRSF2 mutations are found in patients with MDS, CMML, mastocytosis, primarily myelofibrosis, and secondary AML. In MDS, mutations are seen in 12 to 15% of the patients and are associated with a shortened overall survival and leukemia-free survival. In CMML, the frequency is higher, about 28 to 47%, and SRSF2 mutations are associated with increased age, monocytosis, less pronounced anemia, and a diploid karyotype. So our model is the third conditional SRSF2 mutant mouse model following those described by Kim and Con. It's based on a whole locus duplication with a proline 95 to histidine mutation in the first exon of the duplicated SRSF2. And in the presence of Cre recombinase, the FLOX wild-type allele is deleted, and mutant SRSF2 is transposed and expressed from the endogenous promoter. By using various either tamoxifen-inducible or constitutive mouse Cre cell lines, we can achieve either widespread expression of SRSF2, hematopoietic stem cell specific expression of the mutant, or myeloid progenitor expression of the mutant in the presence of Rosa Cre, SEL Cre, or Lysam Cre. And for the latter two, we can combine that with the expression of a yellow fluorescent protein to mark the mutant cells. In heterozygous animals, we can show that we have efficient recombination and full deletion of the DNA of the wild-type allele, and we have also on the RNA level a proper heterozygous expression of the RNA. So when we induce widespread expression of mutant SRSF2 in Rosa Cre mice and follow up with bleeds in a final analysis at 24 weeks, we can see that in the peripheral blood, there is no significant change in platelets or hemoglobin, but there is a significant increase of the MCV with macrocytosis developing after 2 weeks. There's also an increase in the leukocytes and a decrease of the red blood cells. In the peripheral blood at 24 weeks, we also again see that increase in leukocyte number, and we see that there's a significant increase in the myeloid population as well as in the CD4 T cell population. In the bone marrow cells at 24 weeks, we see that there is a trend to reduced cellularity in the bone marrow with a significant increase in the percentage of granulocytes and the percentage of monocytes that is not reflected in the cell number due to the reduced cellularity. But it is indicative of a myeloid bias both in the peripheral blood and in the bone marrow in these mutant cells, mutant mice. In the bone marrow at 24 weeks, we also find that there is a compromised B lymphopoiesis from the pro-B cell stage on, a reduced erythropoiesis, and a reduction in the hematopoietic cell numbers. In the spleen, we see a very similar phenotype with an increase in myeloid cells and a decreased number of B cells, but here there is an increase in the erythroid cell numbers indicative of a compensatory erythropoiesis in the spleen. In the spleen, there is also indications of CD4 splicing isoform changes as detected by specific antibody against CD45RB, with reduced levels of CD45RB expression that are indicative of splicing changes. When we then express the mutant allele in the SCL-CRE, in the stem cells, we see again a very similar phenotype in the peripheral blood with macrocytosis, reduced red blood cells, an increase in leukocyte numbers, all in combination with an increase in granulocytes and macrophages and an increase in CD4 T cells without a significant change in the B cell population. And in the bone marrow at 24 weeks, when we specifically look at the YFP positive fraction, we see again an increase in the granulocytes and monocytes, a decrease of B lymphopoiesis, and a reduced number of long-term hematopoietic stem cells. More importantly, though, these phenotypes are not seen with a constitutive lysyme-CRE activation of the mutant in the myeloid lineage, indicating that the expression of the mutant allele is required within the HSC population to effectively initiate the myeloid bias that we see. So then next, to determine the transplantation potential of mutant SRSF2 expressing cells, we first transplanted activated cells from the ROSA-CRE model into recipients mixed with competitive bone marrow from a wild-type mouse. In these recipients, we saw very low initial chimerism that rapidly disappeared, suggesting that the mutant cells have impaired competitive repopulation potential when transplanted with foreign wild-type cells, confirming what others had previously seen. In contrast, when we transplant cells previously activated in the SCL-CRE mouse, where we used competitors from the YFP negative population from the same donor, in these transplants, we see a long-term chimerism with long-term high levels of YFP expression and an early myeloid bias, suggesting that SRSF2 P95H do have competitive repopulation potential when they are transplanted with H and microenvironment matched wild-type cells. We next looked at non-transplanted mice from both models that were left to H, and we saw that they developed disease 8 to 12 months after P95H activation. They had to be euthanized at this point due to worsening macrocytosis and anemia or dermatitis on the face. In the peripheral blood of these cells, we saw a slight leukopenia, but again, an increased number of granulocytes and a significant decrease in the B cells. In peripheral blood smears, we saw also indications of these plastic cells with hypersegmented and abnormal segmented neutrophils. In the bone marrow, we didn't see any significant change in bone marrow cellularity, but again, a more severe increase in myeloid increase, with also an increase in the immature granulocyte fraction, as seen on the top right corner, a decreased B lymphopoiesis, and again, a reduction in the long-term stem cells and short-term stem cells as well. So, whole non-competitive transplants of whole bone marrow from moribund animals recapitulate this disease, indicating that the activation of the SRSF2 mutation in the stem cells is sufficient to initiate MDS, MPN in vivo. We also analyzed gene expression and splicing changes in stem cells and progenitor cells sorted from the SCL-CRE bone marrow. Here we find patterns of expression that correlate with the observed phenotypes, with downregulation of the expression of lymphoid genes and upregulation of transcripts that are involved in myeloid expansion and/or HSC function. Pathway analysis also identified signatures in the mutant cells associated with human MDS and MPN. In terms of the splicing changes, we saw overall less than 0.5% of change in the total splicing events, but we can confirm the differential misregulated splicing of various genes that were identified, amongst others, differential splicing of CD45RB with a downregulation in the mutant cells, and also compromised misregulated splicing of HNRNAP2B1, TRP53BP2, USP4, all targets of SRSF2 previously described, and here misregulated in HOXB8 immortalized cell lines as well as spleen, but we could not find any differential splicing of ESAGH2. Finally, we also performed whole exome sequencing of DNA obtained from bone marrow and ear tissue from end-stage disease mice as if additional mutations had been acquired. And in the 3 mice that were analyzed, we found mutations in DNMT3A present in all 3 and mutations in PHF6 in 2. TAT2 was also present as a mutation, and further mutations were present in most of the pathways frequently mutated in human MDS and MPS, and also at a similar frequency of 8 to 10%. This indicates that aged mice with MDS MPN acquire additional mutations in genes recurrently mutated in human MDS, but they do not progress to AML. Then in summary, I've shown a new model of SRSF2 P95H mutation with conditional and physiological heterozygous expression of the mutation. And this model recapitulates the core characteristics of human disease associated with SRSF2 mutation on the native conditions, so they do not require transplantations. These key characteristics are myelomonocytic bias, macrocytosis and anemia, transcriptional and RNA splicing changes, myelodysplasia, and additional mutations, again, all key characteristics of MDS MPN. And the data from our model seem to suggest that the primary effect of SRSF2 P95H on normal hematopoiesis is to promote the myeloid expansion from hematopoietic stem cells. So, and finally, I would like to acknowledge Dr. Carl Walkley and all the members of his team at St. Vincent's, as well as all the other co-authors who contributed to this work and the Australian funding agencies that have supported this work. Thank you very much.
So the paper is open for questions.
There's quite an obvious question here, is that what in your mouse model, which is really nice, if you reintroduce DMT3A wild type into it, will you revert the phenotype?
It would be interesting to see. These are all end-stage disease mice, so I presume not, and I presume we have many more mutations in these mice as well, so I don't think that would be an easy thing to achieve. It's, yeah, I haven't actually looked at trying that.
If you did it early on, perhaps it wouldn't develop, perhaps, or you delay development.
Yeah.
It's very, I mean, it's very striking. You have mutations in all 3 of them, DMT3A.
Yes, it did kind of stand out. I have to say, though, that we only had the possibility and the money to analyze 3 mice, so it's a relatively low number.
You could do PCR on the rest and then...
Yes, yeah. No, we've set up more cohorts, and we've obviously collected more samples. Yeah.
So how specific, I'm sorry, I have a question here. How specific you think is the phenotype to that one mutation that you're inducing? Do you feel that you would see similar phenotypes with other mutations, or even possibly with other splicing factors?
I don't think necessarily with other splicing factors. I mean, it has been shown that SF3B1, depending on the mouse model, definitely has a different phenotype in terms of the ring sideroblasts. The other problem is that all the other splicing mutation models have not necessarily been shown to be very actively or accurately reproducing the human situation and patients. So we actually think that this is the only mouse model that accurately represents human MDS without transplantation. But if you can, I think, recapitulate the exact human mutation in a proper mouse model, you probably would find other mutations that induce MDS as such. I think it's just a tricky model for the splicing factor mutations.
But is your implication then that these mutations that do not recapitulate the model in the mouse models are actually not contributing to pathogenesis of MDS? I mean, what's your thoughts on this?
I'm just saying that the mouse models are not always as good as recapitulating the disease, because no mouse model is perfect. And the different constructs and models are based on different techniques to actually introduce the mutation. And for example, the gene inversion technique has been shown to not even achieve a 50% expression of the mutant allele in the mouse model, which obviously in the splicing factor mutations is an essential part of the disease to have the 50%. So there are reasons why not all the models work, and some of that is clear, and some of those reasons are not yet known.
there any other mutations?
Have you seen also some dysplasia of monocytes, like atypical monocytes?
Yes, we did have a hematologist look at it, and she did identify also some dysplasia in monocytes and erythrocyte precursors as well. Yes, yeah. But yeah, clearly the more obvious one are the neutrophils.
Okay, thank you.
Thank
very
you.
much.